In the dense heat of Mumbai, a family of 4 shared a watermelon during a normal afternoon meal. Within hours, the entire family suffered the sudden onset of severe gastric distress. By the end of the day, all 4 were dead. Early reports suggested a tragic but familiar culprit, a batch of fruit contaminated by bacterial pathogens or agricultural pesticides left on the rind. In a massive urban food supply chain, these types of accidents are a persistent, recognized risk. The investigation moved to the city morgue for a routine autopsy, but the standard expectations for food poisoning were quickly overturned. Inside the victims, the medical examiner found something physiologically impossible for a foodborne illness. The internal organs were stained a stark neon green. Bacterial toxins and spoiled fruit do not possess a biological path to alter the physical color of deep internal tissue. The presence of the pigment meant the initial theory of a tragic accident could not explain the condition of the victims' bodies. This led to a new theory. Could a heavy concentration of artificial dye, used to make the fruit look riper, have caused the staining? Under normal conditions, human digestion and stomach acids break down standard food dyes before they can be absorbed into the bloodstream. To bypass those acids and permanently stain solid organs, the concentration of dye would have to be immense. Analysis showed the watermelon contained the substance, but the fruit lacked the physical capacity to hold the volume of dye necessary for this level of discoloration. Achieving this extreme effect required a chemical agent far more potent than any standard dye used in commercial farming. In the toxicology lab, scientists isolated the specific chemical compounds from the victim's tissue, producing samples of the same vibrant green liquid. This visualizes hemoglobin alteration. Certain toxins bind to red blood cells, shifting their color from red to green. Because this altered blood cannot be filtered, it deposits the green pigment directly into liver and kidney cells, remaining trapped in the organ walls. This specific cellular reaction is a unique signature of a narrow class of industrial poisons. The discovery of this reaction shifted the investigation from a medical inquiry into a forensic search for the source of the toxin. With the toxicological evidence in hand, Mumbai authorities officially reclassified the case as a multi-victim homicide investigation. By placing the toxin in a watermelon, the perpetrator used common public anxiety about food safety to create a plausible alibi. The police investigation now focused on tracking the chain of custody for high-potency, non-agricultural chemicals. The crime was designed to look like an ordinary tragedy, hiding the intent behind the familiarity of a household hazard. A crime scene can be staged, and a weapon like a watermelon can be discarded, but the internal chemistry of a victim is much harder to manipulate. Synthetic poisons leave behind distinct cellular traces that are effectively permanent. While a narrative can be crafted to mislead, the persistence of these chemical signatures ensures the body retains a clear record of the events.